Okay, so the next speaker for this session is James Lee, and it's my pleasure to invite him here. He was a Grass Fellow and then a Whitman Fellow with us at the MBL last year, and he's brought a lot of toxins. He's an expert in toxicology, and brought a lot of toxins to the lab, and not just arsenic, but even caffeine. And not just any caffeine, microplastic-loaded caffeine with all the AeroPress stuff. So he's a big AeroPress maker. Fanatic. You should definitely talk to him about this as well, besides his science. And he has this funky worm that he's going to tell you about, which is a relative of C. elegans, but is far more interesting. So over to you, James.
Thank you so much, Alok. And good morning, everyone. Thank you so much for your time. As Alok mentioned, my name is James Lee. I'm a postdoc in the laboratory of Corey Bargman at the Rockefeller University. Today I'll tell you about an unusual protective behavior in the arsenic-resistant nematode, Tokorhabditis tufae. And to begin, I'll start by saying that I love nematodes. And my favorite thing about nematodes is that they're so good at growing in all sorts of extreme environments. So these include places such as the frozen waters of Antarctica, the arid soils of the Mojave Desert, near hydrothermal vents in the deep sea, and even in the irradiated exclusion zone of Chernobyl. And really, worms are great at growing in a whole host of harsh habitats that I haven't even listed here. And we think that this makes them incredibly well suited for studying how animals can adapt to and overcome environmental challenges, such as temperature stress, drought, and environmental contamination. And so with this in mind, the species that I'm studying is called Tokorhabditis tufae. It's a species that I helped to discover back during my PhD in the laboratory of Paul Sternberg. And we discovered this worm from a toxic lake in California called Mono Lake, which is famous for being incredibly saline, very alkaline, and extremely arsenic rich. At an average concentration of 200 micromolar arsenic, that is close to the human lethal dose. And so we were very curious how T. tufae can survive such high levels of arsenic, especially since Mono Lake is part of a growing and global arsenic crisis, one that affects over 200 million humans worldwide. And that's because arsenic is incredibly widespread. Here is a heat map showing all of the locations in the world where arsenic is expected to be present at dangerous levels. And arsenic is released in these areas due to things such as mining, pollution, and agriculture. From there, arsenic can damage the skin, respiratory, reproductive, and neuronal systems of humans and other animals, eventually leading to cancer and death. And so we hope that by studying T. tufae, we might be able to learn about and inspire strategies for dealing with arsenic toxicity. And indeed, we think that we'll be able to learn something interesting from T. tufae because it demonstrates remarkable arsenic resistance. T. tufae can survive prolonged exposure to 30 millimolar of arsenic, which is a concentration that kills the laboratory nematode C. elegans and is equivalent to 150 times the human lethal dose. Now, this next part might not seem related at first, but another thing that we immediately noticed about T. tufae is that it demonstrates an incredibly unique life cycle amongst nematodes. You see, nearly all nematodes lay their progeny as eggs. You can see that in this video of C. elegans. But T. tufae, on the other hand, hatches its eggs inside of its body, gestates its progeny through several larval molts, before finally giving birth to well-developed larvae. At any given time, the mothers can hold up to 18 larvae in their uterus, and these are born one at a time at an approximate rate of 1 birth per hour. And in fact, the larvae that are inside the mother spend a significant proportion of their development in the uterus. So they undergo the first and second larval stages all inside the mother's body before finally being born in a stress-resistant larval stage called the dower. And likely due to its stress-resistant properties, such as having a very thickened cuticle, the dowers are able to survive high levels of arsenic in the same way that the T. tufae adults can. And you can see that in the blue and yellow curves there. And so we wondered whether gestation might be a way to shield the internal progeny from the arsenic in the environment, and maybe this might be a way to give them time to safely develop into that stress-resistant dower stage. And so to test this, I dissected the larvae out from their mothers. And I'm not showing it here, but dissected larvae are actually able to survive and continue their development outside of the mother's body. But what they can't do is survive high levels of arsenic, which you can see in the green curve. So this indicates that the gestating young stages are arsenic sensitive. So I wanted to test whether gestation really does protect the internal progeny from arsenic in the environment. So I took adults, T. tufae mothers, and I exposed them either to no arsenic or 30 millimolar of arsenic for 7 hours. And I found that the number of live larvae in utero was the same in both conditions, indicating that the mothers really are able to keep this high concentration of arsenic away from their sensitive internal progeny. And in an exciting collaboration with Annie Baker, Hugh Harris, Simon James, Elizabeth New, and Liam Adair, we were able to put T. tufae adults under an X-ray microscope in the Australian synchrotron. Now, this is a microscope that allows one to directly visualize elements and where they're located inside of an animal's body at incredible spatial resolution. So when we exposed T. tufae to arsenic and then imaged them, we found that T. tufae preferentially accumulates arsenic in its intestine, which I've outlined in green. And importantly, arsenic is relatively excluded from the rest of the animal's tissues, including the animal's uterus, which is the tissue that lies underneath the intestine. So gestation really does appear to minimize the exposure of the internal progeny to arsenic in the environment. And so we wanted to learn how this unique behavior might have evolved. And briefly, I observed that T. tufae demonstrates remodeled neurobiology and physiology that we think underlies the evolution of live birthing in this species. So first, there is a serotonergic egg-laying neuron that can be commonly found in nematodes, and it's important for regulating the egg-laying behavior. I can find this serotonergic neuron using serotonin antibodies in species like C. elegans, but I cannot find it in T. tufae, indicating that the nervous system has been remodeled in this animal in such a way that we think helps to explain why the animal retains and gestates its eggs as opposed to laying them. In addition, there are egg-laying vulval and uterine muscles in species like C. elegans that contract to open the animal's vulva and push the eggs out of the mother's body. But when I stain for muscles in T. tufae, I see that the vulval muscles have remodeled to form this ringed muscle that runs radial around the vulval opening. And by watching several videos of birthing events, we notice that this muscle acts more like a sphincter that limits the opening of the vulva during birthing and constantly maintains a tight seal between the animal's uterus and the toxic outside environment. I'll also point out that there are no obvious birthing muscles for pushing the larvae out of the mothers, and so I wondered whether the larvae might be the ones that push themselves out during birthing. A lot of the initial observations of the birthing really pointed me towards this idea, and I was recently able to do this experiment where I took adults that were killed by the harsh detergent SDS, and I still see consistent birthing from those individuals. And I'm not showing the data here, but this birthing appears to be driven by activity in the larvae that only begins once the larvae reach that stress-resistant dower stage. In other words, they only become active and push themselves out once they're at the right stage to be born. Prior to this, the larvae that are just developing in the uterus are simply quiescent. And so to summarize what I've told you, T. tufae adults and dowers are remarkably arsenic resistant, and yet their developing progeny are sensitive to the toxin. T. tufae appears to have solved this problem by protecting the sensitive young through gestation in the mother. And this appears to have evolved through the loss of a serotonergic egg-laying neuron, through remodeled birthing musculature, and through behavioral adaptations in the larvae that allow them to drive their own birth. And we're now really interested in investigating how this arsenic resistance and neural circuit changes, muscular changes, behavioral changes, how they all arose in T. tufae by analyzing the animal's genome, which we have recently sequenced thanks to the efforts of our collaborator Taisei Kikuchi. And we aim to test genes of interest from this analysis using transgenic tools developed by our collaborator Ryoji Shinya, whose lab has generated transgenic strains of this animal, like this GFP-expressing worm here. Now, moving beyond T. tufae, we also wondered what other extremophile nematode species we might be able to find in different arsenic-rich environments. And since the arsenic crisis is a global one, we really wanted to take a look at environments outside of the United States, especially those that are close to human communities that have been affected by arsenic toxicity. One such environment is the Atacama Desert in Chile, which is one of the driest places in the world. And similar to Mono Lake, it is incredibly saline and very arsenic rich. And thanks to my collaborators, Andrea Calixto and Gonzalo Pimentel, I was able to join them on a field study to search for worms in the Atacama Desert. And indeed, we found several species of worms that all thrive in the desert. All of them are distinct from T. tufae, phylogenetically speaking, which really highlights to us that there are convergent strategies for surviving high arsenic levels among the nematodes. Excitingly, 2 of these species are growing in the lab as we speak, and so we're very interested in taking a look at their behavioral and physiological adaptations to high arsenic, using our experience with T. tufae as a guide. Now, the last data that I want to share with you has to do with these natural microbes that we also collected from the Atacama Desert. We were really inspired by work that shows that symbiotic relationships with natural bacteria can improve the resilience of animals to their hostile habitats. And while we haven't tested this idea out with arsenic yet, a graduate student in the lab who I supervise, her name is Sarah Omer, performed this wonderful experiment where she exposed C. elegans worms to another stress that's common to the Atacama Desert, which is high salt. And C. elegans is not a salt-adapted animal, so when she exposed them to high levels of salt, close to ocean salinity, she found that the worms quickly die, and you can see that in the orange curve. But when she fed them bacteria from the Atacama Desert, specifically Pseudoalteromonas, she found that their survival dramatically improved. So this is all still very preliminary, but we're intrigued by the possibility that microbes might be able to impart resilience to our animals. And we're also very curious if they might be affecting the animal's neuronal health and behaviors, which we'll be testing next. And so looking forward from here, we're very excited to continue studying extremophiles because we think that they've already solved a lot of the challenges that we anticipate with anthropogenic change. Things like high salt from rising sea levels, for instance, or arsenic toxicity from pollution. We also really believe in studying these organisms in their natural ecology and by working closely together with the human communities that neighbor them. We think that such collaborations can contribute to the science, and we also think that the science can be able to give back to these communities as well. And finally, we're interested in using comparative biology on our worms to identify convergent strategies of resilience, as we think that the organisms might be able to tell us what are the important features of an environmental stress that need to be solved such as perhaps the protection of sensitive young. And so with that, I'd like to thank everybody who made this work possible, including my collaborators, who I mentioned throughout the talk. I'd also really like to thank Corey Bardman, who has been an incredible mentor, and the members of the Bardman Laboratory, who have been wonderful to work with. I'd like to thank my funding sources, which are listed on the right, especially the Kavli and Grass Foundations, who have really supported this work from the very beginning. A big thank you to the Allen Institute, the Kavli and Grass Foundations again, and the National Science Foundation for organizing this event and for their kind invitation. And finally, I'd like to thank you all very much for your time. Thanks.
Thanks, James. We have some questions. I'm going to start with some early career researchers, and we'll get to you. Maybe Yenny can go first, and then I'm sorry, I'm not sure who's sitting behind Yenny, but you can just pass the mic to her, to them once you're done.
Thank you. Yeah, Yenny Voss from the MBL, you know that, but maybe not everyone else here. This is maybe an obvious question, but the other species that you found that are very good in arsenic-rich environments, do they do live births?
Excellent question. They do not live birth. However, they do something that our observations on this are a bit preliminary, but I'll tell you anyways. They lay their eggs in an egg sac. And that's not something that a lot of nematodes that have been studied in the lab do. So these egg sacs are thick structures that have been observed in parasitic nematodes before. These aren't parasitic nematodes, but that's where they've been observed before. And they're known to protect eggs from environmental assault. So there's a striking parallel there between the gestation and the protection of the embryos through these egg sacs.
Awesome, thank you.
Really great talk. My name is Matthew Kapek. I'm from Northwestern. So I'm curious, have you looked at the sensory repertoire of this species? Do you think they might have a specialized mechanism for sensing arsenic?
Totally.
And also, are there populations of tufae that are found like maybe at the edge of the lake or something, or like they have differences maybe in their, maybe an intermediate susceptibility to arsenic or something like that?
Absolutely, totally, thanks. No, that's a direction that we're really excited to go into, especially by using some of the sensitive equipment that the C. elegans neuroscience field has really pioneered in being able to flow different types of stimuli across worms and seeing what their sensory preferences are. I can tell you this, that I've tried some cruder assays taking a look at how quickly can T. tufae sense and appreciate that there's arsenic in its environment. And they don't appear to acutely respond to arsenic. They don't appear to have a fast time scale response to it, but it builds up over time. My guess would be that it's probably sensing accumulated damage over time, as opposed to having some sort of like, let's say, an odorant receptor for arsenic, you know. Yeah.
Okay, we had a couple more questions. Okay, we can start with Amro. Oh, sorry, Stephanie. Okay, I don't know who was asking. I thought Amro had his hand raised a while back.
Okay, so cool talk. I just had a question about the arsenic itself. So presumably free arsenic in the worm is very low, and it's bound to something. So what's it bound to, and how's it moving around the worm?
Perfect. Yeah, so we don't know this in tufae yet, but the X-ray microscopy will help us shed light on this. But we know in other animals that arsenic binds and attacks thiols. It attacks sulfur. We expect that we're going to see a very similar pattern in T. tufae. And in fact, animals and bacteria can use that to their advantage by producing a lot of these molecules called glutathione, which effectively sequester arsenic. And that's been a reported resilience strategy. Yeah.
Okay, we'll do two more questions. There's Martine and
All right.
Laura. We can go with Laura first, and then.
Laura Duval from Columbia.
Hi.
Nice to see you. So is diapause induction different in T. tufae?
Totally. So in species like C. elegans, they can go into dower, but it's not obligate, and it's usually in response to the environment being very stressful, right? Crowding, starvation, high temperatures. In T. tufae, they go through the dower, it seems, obligately as part of their development, which kind of makes sense. If you know that you're going to be born into a toxic lake, they come out ready for it.
Okay, last question.
Yeah, Martin Tresguerres from Scripps Oceanography and ATC. Nice. Great talk. The arsenic part is very cool, but equally impressive the ability to live at 80 parts per thousand salinity. Is there anything known about those mechanisms? Is like ion excretion or osmolytes or anything of that?
No, that's precisely what Sarah Omer and I are interested in taking a look at. So she's really been spearheading the efforts in the lab to look at these animals' salt tolerance. Yeah, thank you.
Cool. Let's give James a big round of applause.